You know that when we eat carbohydrate, or when we eat fat, or when we eat protein, what happened to those that is present in the mostly in our food substance? What happened to those foods? They get digested. When we eat starch, protein, or fat, so when they get into our alimentary canal or the digestive system, they are digested. Digested means the large molecules is break down into smaller molecules. Large molecules are break down into smaller molecules. For example, when we eat a fat molecule, that fat molecule is break down into fatty acids and glycerol in our digestive system. When we eat a protein molecule or protein substance, that is digested into amino acids. When we eat starch, that starch will be digested into glucose. That means the bigger molecule is break down into small molecules. So then for this particular process, it is important to break the bonds present in this molecule because the molecules are tightly bonded inside this large molecule. The molecules are tightly bonded. So these tight bonds need to break and make the smaller molecules. To break the bonds in this large molecule, it requires energy. It requires energy. Using energy, the bonds in this molecule will break. This energy is a very huge energy. Very big amount of energy is needed. But sometimes our body cannot provide that much of energy to break down these large molecules. For example, starch. To break down the starch, protein is done. To break down the protein into amino acid, that means we need to break the bonds among the amino acids in the protein and separate the amino acids. To break these bonds, we need very huge amount of energy. Sometimes our body cannot provide that much of energy because we can't produce that huge energy. Therefore, what happens inside the body? The energy requirement to break these bonds is reduced by the involvement of some other molecule. Another molecule involves to the reaction and reduce that particular energy. Right? So this required energy, the initial required energy to break down the bonds here is named as the activation energy. Activation energy. So most of the molecules, the activation energy is very high. So, but if we can provide that energy, if we can provide that activation energy, what happens? This larger molecule is break down into the smaller molecules. But that activation energy is very high. Therefore, our body may not, because there are many reactions in the body. So then our body cannot provide that activation energy for all the reactions. Therefore, what happens here? Another molecule is involved for the reaction and reduce the amount of activation energy. The required activation energy is drops. To break down this molecule, it requires 100 energy in 100 joules. The activation energy of this molecule is 100 joules, example. 100 joules. But what happens by this molecule? It may be reduced to 60 or 49. The energy requirement is reduced, and then our body has that much enough energy. The molecule will break and then start to make the smaller molecules. So then the energy required by this molecule is reduced by another molecule that is called enzymes. Enzymes. Okay. Enzyme. So normally the molecule which is reduce the activation energy of a reaction is normally is made as a catalyst. Catalyst. Function of the catalyst is to reduce the activation energy of a reaction. Reduce the activation energy of a reaction. So then the enzymes are biological catalysts. Why? They reduce the activation energy of molecules in our cells and our body. Because our body is carrying out many biochemical reactions. These biochemical reactions need very high activation energy. But the involving by the enzymes, activation energy is reduced. Therefore, enzymes are named as biological catalysts. They are biological catalysts which are reducing the activation energy of a biochemical reaction. Biological catalysts which are reducing the activation energy of a reaction. Enzymes. Understood? So these are the enzymes. What is the function of the enzyme? The function of the enzyme is to reduce the activation energy of a reaction. Think that there are two molecules like A plus B. These two molecules need to react to form the compound called C. Okay, let's think that A and B, there are two components in a cell. They need to react to produce the C because we need C for the body function. But the activation energy of this reaction is very high. Activation energy is very high. Then which the catalyst is involving here to reduce the activation energy. What is the catalyst here? Enzymes. Enzymes involved there and reduce the activation energy and let the reaction to happen. Biological catalyst. Enzyme. So when we draw the activation energy in a graph like this, so this is A plus B. Okay. A plus B energy. And to do the reaction, we have to provide high amount of activation. This much of energy required. Okay. And then the reaction happens and produce C. So we have to provide this energy. This much of energy need to provide to react with A and B. This is an example. Like that, when we are providing y-axis shows the energy. So when we are providing this energy level, when this energy level is achieved, that A and B will react and produce C. But what is it's a huge amount of energy. Then what happens when the enzymes get involved here? When the enzymes get involved, what happens? The activation energy is reduced. When the enzymes get involved, the activation this much of energy is reduced to this level. Then reaction will happen. When the enzymes involved there, very easily the reaction can take place and produce the product. Enzymes. So therefore, the reaction inside the body is happening very efficiently and effectively. And other thing is, enzymes are involving reduce the activation energy, but they are not changing the structure or anything inside with them. Nothing happened to the enzyme. They involve for the reaction to reduce the activation energy. After reducing the activation rate, they will leave. Nothing is changed in the enzyme. So we can take the enzyme as it is after the reaction. It is unchangeable enzyme. So the enzymes can be therefore reused because the structure for the enzyme is not reacted or nothing happened to the enzyme. When we put the enzyme into a reaction mixture, yes, the products will produce and enzymes also remains there. The enzymes also remains there. Then structurally, enzymes are not changed after the reaction. There is another important fact about the enzymes. Okay, so remember, what are the enzymes? Enzymes are biological catalysts that reduce down the activation energy of a reaction. And the enzymes are involved for the reduce the energy, but they are not change their structures. They are not participate for the reaction. They are not participate for the reaction. Other things are important factors. Enzymes are proteins. Enzymes are protein molecules. What proteins? They are called globular proteins. They are globular proteins. Their shape is globular. Round shape. Globular. Globular shape. The enzymes are globular proteins which catalyze the biochemical reactions to reduce the activation energy. Right. Enzymes are another effect. Enzymes are globular proteins that serve as biological catalysts in living organisms. Another way of introducing enzymes. Remember, they are proteins. All the enzymes are proteins. Any enzyme is a protein. It's a globular protein. Enzymes are globular proteins. Right. Now, they are about the enzymes. Okay, then we will see how the enzymes get activated or activate another molecule. So these are the general facts about the enzymes. Okay. So what are the major facts I discussed? Enzymes are biological catalysts. Why the enzymes are involved there? To speed up the reaction. Because reducing the activation energy, it is speeding up the reaction. Reaction is speed up. So enzymes are biological catalysts that used to speed up the particular biochemical reaction. They are globular proteins. They are not involved, they are not reacted during the reaction. So nothing changed in the enzyme after the reaction. They reduce the activation energy of the reaction. They are the enzymes. In our digestive system as well as inside in cells also, enzymes are playing a very important role. For example, you know that this reaction, glucose plus oxygen, glucose plus oxygen produce carbon dioxide plus water plus energy. This is called aerobic respiration process. Glucose and oxygen will react to form carbon dioxide, water, and to release energy. The activation energy of this particular reaction is produced by the involvement of the enzymes. Enzymes are involving in these reactors and finally give the product. Therefore, these molecules, the initial molecules of the reaction, are named as the substrate. Substrates. Example, A plus B make the C. So this A, B are substrates, where the enzymes are catalyzed. These molecules are catalyzed by the enzymes. So then the enzymes which are functioning on substrates. And finally, what they produce here, this is the product. This is the substrate. Enzymes are functioning on substrate to make the product. How enzymes are involved in here? By reducing the activation energy and speeding up the reaction rate. Reduce the activation energy and speed up the reaction rate. And finally make the product. But nothing changed in the enzyme. Nothing changed in the enzyme. Then that enzyme can be involved for another reaction also. So therefore, we need very small amount of energy to carry out a particular reaction. Small amount, because it can be reused. The same enzyme can be reused after one reaction. It is starting another reaction. Starting another reaction to catalyze the process there. That is about the enzymes. Right. Is that clear? Then what happened first? Here, how a reaction occurs in the mixture there. How a reaction occurs there. When there is a substrate molecule. There is a substrate molecule in the medium. And there is the enzyme molecule in the medium. The substrate and enzyme together reduce the activation energy and make the product. That is what happened. Substrates, other reactants in the medium. The substrates are there. For example, starch is the substrate. Protein is the substrate. And the starch will combine with an enzyme. Reduce the activation energy. Make the glucose. That is what happened during the reaction. So therefore, in the reaction mixture, there is a substrate. And substrate convert into the products by the enzyme. Substrate is the molecule which binds with the enzyme to make a product. Products are the final result of the reaction. Substrate are the initial molecules. The initial molecules bind with the enzyme and make the products. How this process happens? How these enzymes catalyze the reaction? Catalyzing means reducing the activation energy. How this happens? Now, when we take a particular substrate, think that some substrate molecules have different shapes. Substrate molecules. And enzyme molecules are very important, playing a very important role. The enzyme molecules, inside the enzyme is a globular protein. In the enzyme molecule, there are specific places, sites. There are specific sites in the enzyme molecule which let that substrate molecule to bind. There are specific sites on the enzyme molecule where the substrate molecule can attach, bind. In the enzyme, when we take the enzyme molecule, here, in the enzyme molecule, there are specific places where the substrate molecule can be attached. Those specific sites or places are called active sites. Substrate molecules are binds the active sites of the enzyme. Okay, it's like this. Think now, that means how the substrate binds to the active site, because the shape of the substrate molecule is similar to the shape of the active site. That is why they can bind it. Otherwise, they cannot bind. How now? See that yeah, I will draw. Is the enzyme molecule there? Is the enzyme? These are the active sites. Here we have the active site. This is the active site. This is another active site. Therefore, the substrate should have the similar shape for these active sites to bind to it. The substrate should have a particular shape. So these are substrate, these are enzyme molecule. These are substrate molecule. So these are the active sites. Active sites. Then the substrate molecule, they have active sites to bind to this particular enzyme. Substrate molecule have specific places which can be bind to the active site of the enzyme. Now what happens? This particular the substrate and the enzyme are get bind with each other. And finally, it makes the enzyme substrate complex. It makes the enzyme substrate complex. Enzyme substrate complex. The substrate molecule binds to the active site of the enzyme because shape of the substrate molecule similar to the active site of the enzyme. It binds there. Whenever shape is not the shape is not the substrate. They can't bind because shape is different. No, they can't bind because the active site shape should be similar to the substrate. So these molecules cannot bind. Enzyme substrate complex. And what happened after making the enzyme substrate complex there? What happens now? Now this is the enzyme molecule. The substrate molecule with enzyme substrate complex. This substrate is break down into smaller molecules because of the reduction of the activation energy. Okay? It will be break down This one will be break down into different components. And find the one they will release. They will release. These are the products. First what happened? Enzyme binds to the substrate molecule and produce the enzyme substrate complex. Reduce the activation energy of the molecule and then break down into products. Products released, and what happens then? Products will release because now the shape of the products are not similar to the active site. Only the substrate shape is similar to the active site. When it breaks down into smaller molecules, the product shape is not similar to the active site. The products will leave, and enzyme remains as previous. It can start another reaction. It can start another reaction. Okay, this is how the enzyme reactions are happening. The process of enzyme reactions. Remember, so therefore here only a particular substrate can bind to the enzyme. Not all the substrate can bind the enzymes. That happens. Because only the similar shape substrate molecules can bind with enzyme. Only specific molecules. Enzymes are specific for the substrate. Therefore, we can make that enzymes are specific for the substrate because only selected substrate bind to the enzyme. So therefore, we use the term enzymes are specific, substrate specific. Because the enzymes we use to digest starch cannot be used to digest proteins. The enzymes which are used to digest lipids cannot be used to digest the carbohydrates. Because enzymes are specific for their particular substrate.